Hello everyone and welcome to our Astro stream. We are doing course with Reut about astrobiology, and today she is going to talk to us about the extremophiles. I'm very excited to hear about this, and the series is going really amazing. Without further ado, I am gonna shift to Reut, and Reut, you can go. You're on the screen.
Thank you very much. Hello, everybody. It's great to see you back on board for our precious few remaining lessons. So I want to thank you again for coming and putting in your time and your brains into our mini series. It's been a terrific experience for me and for Shweta. So I'm going to talk a little bit about what we did before and what we talked, and everything is connected in astrobiology. So stay tuned and stay till the end and have a great time asking me questions afterwards. So I'm going to share my screen now with my presentation. Great. Cool. So if everything is okay, right? Okay, so astrobiology in the known universe. So astrobiology centers about a lot of issues, and the center of astrobiology is life, how it originated, how it was created, what is its distribution, how did it change our planet, where are we going to find more life? And the human race has been pondering about this issue for many centuries. And I just want to reiterate that till now what we talked about so far in our series is that we talked about the origin of life and their evolution and the issue of time scale. How long does it take to create life? How long does it take life to evolve? What did Darwin think about evolution? What did Lamarck think about evolution? What are the atomic and common elements in the universe, the atomic and elements which are common in our human beings, in all the biosphere on planet Earth? What is the primordial soup? What are the differences between the different theories about the origin of life? We have RNA world, we have lipids world, we have panspermia, we have a few more. We discussed that in our past YouTube, which you can now look at the websites and you can check it out as well on YouTube with AstronEra's help. And today we're going to talk about extremophiles. And the reason extremophiles are so interesting is that first, it's a relatively new thing for us humans to understand how extreme life can be and could be living very happily in very weird conditions. And also I want to say today that we really like microorganisms, and especially the extremophiles from them, because what we find out is that in generally we are one with microorganisms of planet Earth. They are in our gut microbiota. We use them for drinks. We use them for food, of course. We use them now for medicine, so to keep ourselves healthy and happy with antibiotics and other types of medicine that we first were discovered from microorganisms and sometimes fungi. And what's also interesting is that when you find an extremophile, a microorganism which is happy in extreme environment, they usually have a lot of mechanism. Sometimes part of those mechanisms are pigments or proteins or something else. And when we figure that out, we can actually take that from that microorganism. We can culture it, we can use it in the industry, and we can put it in a very sterile fashion in things that helps us, whether it's UV, a resistance to radiation, for instance, resistant to UV, and then we take that protein, we take that pigment, and we put it, for instance, in, anti-sun and radiation serums. Astaxanthin supports eye health, is something that it's a red pigment that we actually take from microalgae, which produces. So we're really happy to find all these different solutions that comes from extremophiles. Sometimes it's enzymes that can work at very low temperature, and then we can use them as, to disintegrate all sorts of detergent and things like that when you're using your washing machine. So it's a really good avenue for biotechnology. I'm still voting for learning about our world just to do basic science, not just because it can be applied in biotechnology, but there you are. And here on the left screen is one of our most significant my extremophile organism, which is a tardigrade, and we're going to talk about it later on in the conversation. Stay tuned. So, can life walk the walk everywhere? If you were here last time, then you saw this phylogenetic tree, which composite all the life forms that we know that we have been able to sequence their genome and their DNA. And every red dot here in this massive tree, okay, basically represents additional 100 to thousands of different species that are part of that specific branch. And it's a staggering amount of life forms everywhere, of species, of taxonomical groups. And we want here today to talk about extremophiles. How much of that huge tree is in extreme environments? I mean, what kind of extreme conditions are we talking about? So we're talking about temperature, which could be very high, could be very low. We're talking about pH, the level of acidity or alkalinity. We're talking about atmospheric pressure. So most of us are walking, talking, and working today, at sea level. That's about 1 atmospheric pressure. But today we know a lot of things that are happening actually beneath the ocean and very deep in the ocean. And most of our planet is not land, is ocean. So perhaps we have much more life forms underneath the oceans. Who knows? We talk about salinity. What does it feel like to live in 50%, salinity solution, or someplace like in a salt crystal, and find what kind of mechanism do we find with microorganisms that are able to do that. Metals are usually toxic, but we find extremophile that can do that. Low water activity desiccation, hyper arid places like deserts or Antarctica, which don't see a lot of water available for the organism. That's what it means when it says desiccation or low water activity. It means that the water is not available for the microorganism or for the species or for the life form. And for us, we're like 17% water. What happens to us when we don't have water? We die. There's some microorganism out there, in tardigrade, which is not a microorganism, just a microscopic one. It's like a great example for something that can live exerting about 60% of its water, which is amazing. It's like a type of hibernation, and it's amazing that it can do that. But not a lot of bigger life forms on planet Earth can do that. But we do find microorganisms which are available that are able to withstand this part. And we're talking about radiation and what happens when some microorganism from planet Earth or a life form, what happens to us when we see radiation, when we meet radiation, what kind of levels of radiation can a human withstand, can some microorganism withstand? So it's a very interesting talk today. And seriously, this is just a background, very simple background conversation. So feel free to dig in more on the internet, at NASA website, astrobiology website. Wikipedia is not bad at all. And also Google Scholar has some terrific reviews. So moving forward, what are the physical problems with some of these extreme? Okay, it's an extreme environment to work under, 10,000 atmosphere or work in 100 Celsius degrees. But what actually is happening from a physical point of view? And every extreme environment has a physicality to it, which the biological entity has to respond to with some mechanism in order to survive and in order to grow. So for instance, we're now starting with the issue of temperature, okay? So when you see here on the left side, you see it, I hope, they're basically upping the temperature a little bit in this cute animation from a wonderful website called, and you can see the balls, which represent here atoms and molecules, they're starting to move around much, much quicker and all over the place. And this is what happens when you rise the temperature in general. That the higher the temperature of a substance, the greater the kinetic energy of those particles. And particles could be atoms and could be molecules. And if you think about our body and about our enzyme inside our cells, everything is connected to the fact that you have molecules doing interaction with one another, okay? And those molecules are made out of atoms. If I bring up the temperature and I bring more kinetic energy to the atoms which are building a specific molecule such as a protein, I'm causing that protein to disintegrate, to lose its structure, because the atoms are all not sharing their electrons anymore, and they're starting to pull apart, they're starting to move around. How do you cope with that kind of so much energy going into the particles? That's one problem with temperature. The other problem is that on the sea level surface, we have this, not only on sea level surface, but we have this cycle goes on between materials that goes between the solid state of water into their gases and into their liquid. And what's happened with temperature, whether it's a high temperature or a low temperature, is that you take into consideration and you're changing the structure of the water molecules. The more solid they are, the more rigid they are. You cannot move them around. Also, water is one of the few substances that we know of that when it freezes, it actually expands from a surface point of view. It doesn't go smaller, it actually expands. And that's a very big issue for stuff that live in a cold area. Because if 70% of their body weight is in water and they go into a freezing place, then what's happened to the water in their veins, in their cells, and all that area? It expands and it ruptures. It ruptures the cell membrane, it ruptures the blood vessels, it does a lot. Those ice crystals are a big issue. That's why one of the most brilliant things to do is actually to exert the water out. When the body feels like an extremity coming on, when it's going into a freezing mode, tardigrade, they exert the water out of their body, and that way they lower the chance of being hurt physically by ice crystal. But this cycle between solid, gas, and liquid of the water molecule, which is a very basic thing on planet Earth, right, is something that changes which changes, of course, with the atmospheric pressure, okay? With the atmospheric pressure, you exert this energy on the molecules and on their structure together. So obviously, the transition between solid, gas, and liquid states will change depending where the molecule is and how much pressure, atmospheric pressure, is put upon it. Now, another thing, like I said, is that when you have the winter ice, when you have an ice crystal, it expands, right? It gets very rigid. So things tend to really they get very delicate, and things are getting mechanistically, they're getting ruptured, they're getting torn. So it's very interesting to see that, for instance, in some plants, they have membranes which can handle this type of situation, but most organisms, if you put something in the ice, you can break it off very easily. Everything becomes rigid, it loses its flexibility, and our body cannot cope with that at all. Like, human body cannot cope with this type of freezing at all, and we tend to lose limbs when we get to the freezing point for too long. So be careful with that. And tardigrades have a great way, like I said before, they exert the water out, and that's why they can actually survive very cold temperature, like big minuses in space. Let's talk about like a really nice psychrophilic. So psych, it means cold, and phil means from philos, from the Greek word of love. It means loving the cold. And we have hyperpsychrophilic, which can go below -20, and we have psychrophiles, which are adapted to cold temperature around 0. But when you have psychrophilic, you have between 10 to -20 organisms that can really, really survive these harsh conditions, and they can survive, and they can reproduce, and they can grow, and everything is going well. So what you have here on the left is a cute example of a Antarctic midge, which is an insect, and it can survive in temperature below -15. And one of their mechanisms is, aside from having a complete genomic adaptation to it and proteins which are able to withstand the cold, they also burrow to a depth of about 1 centimeter. And what happens is that below the surface, you have a very stable niche that the temperature, because you are surrounded by a solid phase, you're surrounded by soil and by the regolith, that actually keeps the temperature very, very stable. And they're not that cold, they're not that exposed to the wind or to the difference between night and day. So that helps them to be in a very specific niche with specific type of temperatures. It accumulates pteralose, glucose, and erythritol. So pteralose and glucose are types of sugar. Now, I'm sure perhaps some of you would know is that when there is an osmotic pressure happening, that if you have too much, if there's too much, for instance, particle outside of your body, water will tend to go to where most of the particles are. That's why, for instance, candy never gets spoiled. It doesn't have any bacteria growing on it because there's so much sugar in there that anything that tries to survive in a candy basically loses all its water to the candy and it cannot perform well. So when you see this type of adaptation, and we see it also in, by the way, extremophiles, which live in saline, highly salinity areas like halophiles, Then basically what happens is that you see that inside their body they have these big heavy molecules. It could be sugar, it could be other types of molecules. They're pretty big. And what happens is that the water has no, if anything, the minimal water that they find around them in liquid form, they would like to go and stay within their own body and not run out, because that's how you prepare, that's you sort of like, it's a mechanism in order to make sure that the few water molecules that you do have in your body, they won't try and go away to the environment. And if you do encounter liquid water, and water is available in sometimes these very arid area which are frozen, then you will be able to accumulate them. There's all sorts of membrane and shifts in protein structures and type. So for instance, remember I talked to you about how rigid things become. So basically what they do is that they bring in unsaturated fatty acid into their membrane. So think about like a drop of olive oil in your fridge versus a butter. So the butter will not freeze, but a butter will stay very, very rigid and will solidify. But you will still have some viscosity, you will still have some thing moving along with the olive oil drop. And that's because it has unsaturated fatty acid. It's a very long molecule, and it has a lot of carbons within. What happens with that is that even though it's really, really freezing, it's still a very flexible membrane, still very flexible molecule. And if you have a membrane which is built out of these flexible molecules, you're able to maintain movement, which is super important in very cold areas. And what we see is, like with other psychrophiles, is that they're super adapted to that temperature range. If you move these organisms from their temperature range, for instance, research found out that if you take these Belgica antarctica and you move it into 30 degrees, eventually within a few hours they die. They don't have the right proteins, they don't have the membrane structure to cope within a few hours with this completely new temperature regime. So that's a very interesting thing to look at in the extremophile world, in the cold world, in the Belgica antarctica. Most of the psychrophiles we know today are actually microorganisms. Now, if we're looking to the other side, to high temperature, then we have mesophiles, thermophiles, and hyperthermophiles. And they're very interesting organisms which have been indicated more than at least 150 years ago. But we actually have been able to really nail down and isolate a few important bacteria starting from the 70s and the 60s of the last century. And what you see on the left side is how Thermus aquaticus, which is an extreme thermophile that was isolated from Yellowstone in the 1960s, and how does it look under an electron microscope. And on the right side, you can see how this environment looks like. It's very colorful because you have not only a range of because you have a range of minerals that's coming from the depth of the earth and surfacing with hot water springs and everything. But also you have a very interesting different regimes for different types of bacteria, cyanobacteria, and so forth. And Thermus aquaticus was very, very important later on in our work because it was able to live happily at 50 degrees and survive until 70 degrees Celsius. And we found out that it has many enzymes that are completely adapted to that range. It also has, for instance, a DNA polymerase. I'm hearing something on my phone, and everything's cool. And what I wanted to say is that Thermus aquaticus, in short, TAC, has entire system of proteins which are adapted to high temperature. And what does it mean, a protein that is adapted to high temperature? So remember that I told you there's a lot of kinetic energy that comes from the temperature regime when you have high energy coming into these molecules and into this atom. So first of all, one of these mechanisms is something which is called heat shock proteins. And heat shock proteins are in abundance in Thermus aquaticus and other thermophiles and hyperthermophiles. They are short string supportive, basically chaperones. They're heat shock proteins, and their only role in life is to come to other proteins and make sure they do not denaturate. Okay, they don't lose their structure. So they come, they hold it, or they go on top, or they just hold the molecule, the target molecule, and they make sure that it does not denaturate because of the temperature scheme that's going around. Another thing is that they have an abundance of heat shock proteins. It's not just one, it's not just 10, it's like hundreds of heat shock proteins living within and helping them to cope with that. Additionally, we find in thermophilic proteins, we find that they change the sequence of their protein. So a lot of the proteins have atomic and have van der Waals and have hydrogen bonds. And what we do find with thermophiles is that they have much more, let's say, stronger connection. They have more compact, close together atomic connection, more van der Waals connection. I'm not going to go into that. But basically, the more atoms you have that can share more electrons between themselves, you're going to need a higher energy in order to break that apart. So we see a lot of signature happening with extremophiles, and we find today extremophile mostly, of course, in next to volcanoes, also in like you see here in a lot of thermal springs. They're fascinating creatures to see, and they're also fascinating creatures to work with. Just imagine you're going to need like a cooking oven in your lab in order to make sure that they survive in the degrees that they want to. You're going to need an incubator for 50 degrees, for 70 degrees, and perhaps even higher. One of the scientists that his name will ever be connected with Thermus aquaticus intact, his name is Dr. Kary Mullis. And Kary Mullis won the Nobel Prize for the polymerase chain reaction. So the polymerase chain reaction, it's a molecular tool. I discussed it a little bit, and some of you know it already, actually, from your own studies in our former discussions. And what Kary Mullis has found out is that in this thermal process of PCR, you need to reach to a very high level of degrees, not a high level, you need to get to high degrees in order to make sure that the double helix of the DNA starts to denaturate, starts to open up. And it's usually around 95 degrees. And what Kary Mullis has found out is that this Thermus aquaticus, this thermophilic bacteria, actually has the right DNA polymerase that even at 95 degrees can still work. So you can up and make the, for instance, starts the DNA double helix to separate. And then you can just lower it just a little bit, perhaps to 72, perhaps to 76 degrees. And all of a sudden, you can have the TAC, the DNA polymerase, working again on the DNA, making sure that it's hitting it according to the nucleotides that you supply it and the primers that you supplied it in this PCR process. So because he sort of found out that he can use this DNA polymerase from a Thermus aquaticus, so Kary Mullis got the Nobel Prize for chemistry, if I'm not mistaken, in 1993. And he's still a very active scientist. You are more than welcome to look at his, I think, at his labs and things like that. Very interesting fella. And it's just to show you, when I was talking about biotechnological application and basic science, this is a biotechnology application coming from the TAC polymerase and from something that we found in the 1960s in Yellowstone. And what else do we have here? So going into... Yeah, is that the one? Yeah. Going into the deep, deep ocean, basically what happens, it's a pressure problem. Big pressure problem. Now, what we find life forms centering in the Mariana Trench, very deep, some 5 kilometers deep. That's like 500 atmospheric pressure. Sometimes we find things even deeper at 11 kilometers depth today. And that brings us to around 1,000 atmospheric pressure. That's huge amount of pressure on anything to survive. And what Dr. Julie Huber here says in 2013, and she goes and she explains that in 1977 was the first time scientists looked at the ocean depth and saw life. Till then, they couldn't realize that there could be life at all in this extreme deep ocean. So let's just listen for a moment.
But they saw this amazing bounty of life.
I hope you can hear it.
Cannot hear it now.
That were going on that in the submarine. My sub called up on the radio and said, I thought the deep sea was supposed to be dead. Why am I seeing clams? I mean, it was that unexpected. And it was a few years later.
Yeah, it's better. Can you start it from the beginning? Because I think the earlier part got.
But they saw.
1, 2, 3, starting. Oop. Where is I? Here it is.
8 was no ocean explorer. but they saw this amazing bounty of life on the seafloor. And in 1977, everybody still thought that basically the deep sea was dead. And if you listen to the original Allen communications that were going on down in the submarine, one of the scientists in the sub called up on the radio and said, I thought the deep sea was supposed to be dead. Why am I seeing clams? I mean, it was that unexpected. And it was a few years later when Colleen Cavanaugh and others figured out that these tube worm guts were packed with chemosynthetic bacteria that are oxidizing sulfide.
Okay, so back to our place. What are the physical pressures? What is the basic problem that life have to work with when they're in the bottom of the ocean? And just didn't think anything's gonna be there. So first of all, us scientists, and I would be the first to admit it, we not necessarily have the most vivid imaginations, and we work with what we know. And what we knew till the 1970s is that basically you need the sunlight for energy, or you need, I don't know, other type of energy like lightning or things like that. At the bottom of the ocean, we couldn't figure out what would be an energy source for life form. And what happened is that this crazy, marine engineer said, no, I'm going to design a submarine that can go so deep that nobody has seen before. And he designed Alvin. And Alvin, since 1965 or so, has been working in the ocean. It was retrofit like a few years ago. It discovered like 300 new species in the depth of the ocean. And it's a miracle. Today, 3 people can go down in Alvin and take a look. But even more today with NOAA and other organizations in the United States, there's a new generation of rovers that are able to withstand even further down. And what we realize is that there is an energy source, and that energy source is actually underwater volcanoes. And all the magmatic interaction between the continent is happening not only in the Himalayas, but it also happens underneath in the depth of the ocean, in the Atlantic Ocean and the Pacific Ocean. In these areas, the magma is allowing for a lot of nutrients, to come out from the crust of the ocean. And you see magnificent ecological things which are in complete darkness and sometimes are very, very cold. The hydrothermal vents themselves are super hot, okay? They can be really, really hot. They can be like 400 degrees, 500 degrees Celsius. But very quickly around them, you get to really deep ocean cold, and it's very dark. And you see life forms. So it's amazing how persistent life can be and how they can arise in places which we scientists don't necessarily realize. But let's talk about specifically what are the physical problems here that we have with pressure. So with pressure, what happens? It happens 2 things. First of all, you because of the pressure, you perform a physical stress on the molecules. And again, you get this denaturation problem that we saw, for instance, with the high temperature. Okay, so what happens is that you see the quaternary or the big structures of the protein, they start to be separated to their what we call heteromers, or their different parts. And then the different parts, each within themselves, they start to disintegrate and denaturate to their secondary and eventually to their first, secondary, and primary structure. Basically what happens, the more pressure you put on a protein, it disintegrates and it doesn't work anywhere. But we know that life encompasses also membranes, also encompass DNA, also other things. So what happens to those things when you use a lot of pressure? So surprisingly, pressure works a lot on hydrogen bonds. And apparently, for instance, with DNA, the stable helix is very stable even under a lot of pressure. So for instance, the DNA will not denature under a high pressure. Enzymes will. Membrane will feel the pressure, but we still haven't figured out what is exactly the mechanism with which that type of membrane can sustain a lot of pressure. But again, we're talking about microorganisms and things that would be able to work in such an environment. What do we see, for instance, in the deep ocean? What do we see, for instance, in the deep ocean? Okay, we see something which is a single cell with a lot of nuclei. It's the stuff on the left side of your screen. It's called xenophiophores. It's huge, actually. It's between 10 to 20 centimeters. It's not a microorganism at all. it has this structure around it, which it borrows from the nutrient around and makes its own shell. And it feeds, and it's very happy down below. It has very unique enzymes, but that's a very big living thing that we find there, amongst other things. And just around the hydrothermal vents, be it a black smoker or a white smoker, around the Mariana Trench, we find these clamps and shrimps and limpets, and they're all very, very tightly together. They're living one from another, and they're all very happy because the pressure, because it's, how should I say it, the high pressure which they live in the depth, which is about 700 atmospheric, is balanced because of the balanced with the high temperature in which they live in, right? So they're living next to a hydrothermal vent. It's like a live active volcano. And that live active volcano is very, very hot. But because there's so much pressure on the molecules and there's so much temperature coming up, they're both able to sustain one another, I think. And what we find is that we find a lot of bacteria are chemoautotroph and heteroautotroph, are very happy to live within the mussels, within the shrimps, and they're happy to work in that environment. And everything is happy. It works around 700 atmospheric, but it's still very much alive and reproducing everything. And these are important for us to understand, for instance, when we compare what's happening with Venus, which was in the news recently, right? So it's interesting to understand what's happening in the depth, in high temperature, but also in low temperature, right? And then see whether something of that is happening also on Venus, which has been in the news. So we have discussed just a bit. We discussed just a bit about the temperature extremes of psychrophiles and thermophilic extremophiles and how we were able to isolate and work and understand what type of mechanism they're using. We talked a little bit about atmospheric pressure. And I have to say, this is all very active research. There are new findings every week, every month. You're more than welcome to check it out in Astrobiology magazine or otherwise. It's a fascinating area of structure, of research. And it also requires very specific instruments and very specific pressure, pressure vessels and how to work in the ocean. So it's a fascinating area to go into. I want to talk now a little bit about the radiation. What's the problem with radiation? And the problem with radiation is that we have ionizing radiation and we have non-ionizing radiation. Ionizing radiation is pretty simple. It's like X-ray or gamma rays. Basically, you have things that are coming straight from the atom, radioactive, sometimes it's radioactive, and basically it's neutron or electrons or protons, a particle that comes in such a high energy that it breaks the atoms that it encounters and creates ions. So what happens with X-ray or gamma rays is that our DNA gets sometimes a double strand break or sometimes a single strand break. So we have this helical shape and all of a sudden it breaks at some point. Now, life has evolved on planet Earth. They know at one point they're going to have this type of X-ray or gamma rays because we know of gamma burst radiation coming from galaxies around us and stars going supernova. So it's not a completely rare thing that happens. And the DNA has its own correction facilities. It has its enzyme that know to recognize there's a break. There's enzymes that know how to fix it and so on and so forth. But still, if you have too much rays, and for human species, it's enough to have 5 grays. That's the measurements of radiation that we use today, the grays. So if you have 5 grays for a few days, you die basically because your entire DNA is completely chopped up beyond opportunity to fix it, to correct it, to make the cell work again. And that's very, very bad. And that's one of the main problems astronauts will have to deal with on their way to, for instance, Mars. A long journey in space means a hell of a lot of X-rays and gamma rays and ionizing radiation that can cause them basically to non-function and die. Non-ionizing radiation, specifically, for instance, UV light, brings a lot of energy, but doesn't completely break the double helix, doesn't cause a break, but it does cause some switching of electrons. And because of that, there's a dimer that's formed between 2 bases of thymine. It's called the thymine dimer. And that causes the DNA to cannot be translated, cannot be transcribed, and sometimes can cause a lot of trouble. So basically, if you change something in how the DNA looks like, And if you recall from our former conversation, we were talking about how there were triplets of letters of the DNA, that from those triplets we knew which amino acid, and those amino acids eventually created our proteins, which are the basic necessity for a cell to function properly. So if you change any codon in the DNA, you might either not change anything, for instance, TTT and TTC is still phenylalanine, but you might actually cause a shift in how things are read on the DNA. And you might actually cause for a very different amino acid to form in a very critical space in a protein and completely block that action and cause death or something. So one of the things that are happening with UV radiation is that if you collect too many dimers, there's no way to correct it, and some other things happen that cause eventually for cancer and for other problems. So that's the radiation. That's the most basic problem life forms on planet Earth have with radiation. It has to do with changing the genetic code. That's how we see it today. And how extremophiles are able to resist radiation. So first and foremost, they provide themselves with a lot of coating and certain types of molecules. In this case, it's a pigment called skytonim. And skytonim absorbs UV radiation and other type of electron. And the pigment itself is able to get that radiation and, for instance, extract some electrons. And by harnessing that radiation and extracting electrons, pigment basically is getting most of the hit instead of the DNA. So if you have a lot of this, it's like your first line of defense, and you're avoiding the UV radiation and other from hurting the DNA. Another mechanism that was found with Deinococcus bacteria, which is quite an interesting bacteria, it's that we found it on nuclear, so completely bombarded by gamma radiation and radioactive decay. But we found out that this specific bacteria is really, really good, like Superman good at fixing its DNA and working with DNA which is very much fractured. So that's quite an amazing thing. So they have a lot of enzymes, much more than we have in our own cell, that their only function is first to find out where the breaks are and then start constantly to fix it and fix it and fix it and fix the DNA properly so nothing bad will happen to the cell. So it's a big mechanism, and it's a big investment from a bacterial point of view, but that's what makes it survive this type of radiation conditions, which is pretty much cool. And tardigrades, which are water bears, are eukaryotic organisms. They're about 0.5 millimeter to 1.5 millimeter organisms, and they were found to be absolutely superheroes in space. And you can find there's thousands of strains of tardigrades. You can find them in a lot of different ecological niches on planet Earth. You can find them in deserts, in Antarctica, in swamps, on wood, detritus, and things like that. They're very happy creatures, and they're very, very cute. You don't need a big microscope in order to find them. And what we realized is that they're able to withstand super low temperature, like minus 272 up to 150 Celsius degree. They can go without water for at least a period of 10 years. And they're extremely resistant to radiation. So tardigrade, or a kleine Wasse, by the German researcher who also found them in the 19th century, are basically heroes of the extremophiles. They can withstand different types of extreme conditions, and this is mainly probably due to their capability of exerting water. Once you don't have water in your system, you are prone to radiation on one hand, but you're not prone to all the processes that what happens when you don't have water in your system, and nothing works. Once the water is out from a tardigrade, it looks like dead, but it's basically hibernating. And when we say that they are able to withstand temperature, we mean that after we resurrect them from their hibernation, they are able to reproduce, they are able to grow, they are able to have healthy offsprings. And that's how we measure how something is able to withstand these conditions. We don't know fully how they protect themselves till the end. We have no idea how, from having no water, all of a sudden having a few molecules of water, how does that work within their body to signal them that, okay, now you're back into normal normal time, and you can start looking for food. It's a very complex microorganism. Again, sorry, it's not a microorganism. You can see it with a really small microscope, 4x, but they're cute, I have to say, very, very, very cute, and they sent to space, and people have seen that they were able to withstand different conditions in space. So they are the most complex animal that we know that can withstand several extreme conditions. So before we finish and we talk a little bit about Venus, just wanted to summarize and put some thoughts and say, okay, so the extremities that we now know are mostly defined by our human perspective and by our own eyes. It's quite obvious that we can find an unimaginable place, right? Scientists weren't really aware that stuff could live deep in the ocean and like in such high pressures and low temperatures and things like that. I mean, finding a bacteria in a nuclear reactor is like mind-boggling. Why would anything would be able to withstand the harsh physical reality of a nuclear reactor? And also, I haven't spoken about salinity, about what are the mechanisms over there on how to survive salinity, how to survive acidity. But I can assure you that we are slowly understanding the mechanism behind each cell's strategy in order to withstand its condition. Life really want to survive, and they are very flexible in the way they do it. They have a lot of mechanisms, whether it's the genetic code, whether it's in the membrane, whether it's in their ecological niche. You know, some bacteria live within the rock. Sometimes they live deep inside a rock. Sometimes they live within cracks above the rock. Sometimes they burrow, like we saw with the Antarctic Midge. So life, not only from the biomolecular point of view of them, but also from how they interact with the world, they are able to find a niche which is stable enough for them to survive and to grow. So it's super interesting. What are the possibilities out there? So I haven't spoken about everything. If you're really interested in a full-on course that will take you several months to finish, but it also has a free option, then you can go to Coursera. And my dear friend, Professor Charles Cockle from Edinburgh University has an astrobiology course there, which has a lot of goodies and have a lot of information about these different things and different issues altogether about astrobiology. And with your permission, we'll talk a little bit about Venus because it was in the news today. So what's the thing about Venus, and what is phosphine, and how about life on Venus? What's going to happen then? So this is the list that I found about all the missions that went to Venus in the last, 40 years, maybe even more. Yeah, 40 years. You can find it on a NASA webpage. And basically, most of them were orbiters or flyby mission. The best one that I found, really nice photos, were from the Magellan, which worked until 1994. It was a radar mapping mission. And we got really good information on how the surface of Venus looks like. And the Venera mission was the only one, the only mission. It was flown by the Soviet unit at the time till 1983, and it was the only mission that actually was able to really land, cross the acidic layers of the clouds, go down to the 500 Celsius degrees and take photos. So some of the photos you're going to see are from the Venera. This is by the Magellan mission. What you can see here is a very high mountain, 8 kilometers high. It's called Ma'at Mons. The average temperature on Venus is 464 degrees. It can get quite higher than that, up to 700, 750. Its circle around the sun is mostly like ours. It's 224 days per year around the sun. We have found out that it has volcanic activity on the ground as well. So you have this energy going on, and probably a lot of geological feature coming from the heart of the planet, from a magma, and going onto the surface. These are some of the photos that Venera 14 took many, many years ago. And you can see it looks a bit rocky. It looks a bit clayish. There's a lot of layers in the rock there. What you see is also a part of the lenses that Venera had, and so on and so forth. We know it has a lot of acidic clouds, specifically.
Sorry about that.
It was fun. sulfuric acid clouds atmosphere. There's a lot of organic molecules that we know that are now in Venus atmosphere. There's a lot of there's a lot of water molecules, carbon dioxide, we have helium, we have CO, we have monodioxide, carbonate, we have HCl, which is an acid basically, hydrogen chloride, we have argon, and we have other molecules as well. So there's a lot of rich things happening in its atmosphere, but it's very, very acidic. Like we think it's between 1 to 3 on the pH scale, which is super acidic. This is a color photo from the same, from Venera 13 image, just to give you a look at how it looks. It's kind of reddish hue and everything. I mean, in the Venera images, the Venera, once it landed, it had maybe a few minutes, maybe 20 minutes before it stopped transmitting. It's such hard conditions for electronics, and for everything. Can you imagine sending something today that would be able to withstand such conditions? That would be quite a thing. And talking a little bit about the phosphine discovery that was discussed last week, it's an astronomy research group from Cardiff, and they did a really nice job from 2 independent, I think, observations with different telescopes, and they realized there's quite a lot of phosphine in the atmosphere, in the clouds of Venus. And they didn't say anything about finding life or anything like that, for sure. But what they did say, they said, look, we looked at the chemistry of how phosphine is being created and how we can do it in the lab, but we couldn't find any way that is relevant to the conditions that we've seen on Venus. So we think either it's a completely new chemistry that we're not aware of, and that's always a really solid possibility, by the way. And the other thing they said, maybe it's something that life has produced. I can hear someone. And so on life produced. So basically, phosphine is known from planet Earth. It's a chemical that a French chemist found in the 18th century, if I'm not mistaken, and it's very, very toxic. The idea that it's perhaps by some sort of a life form in Venus has perhaps produced it, it's a remote possibility in my mind. It's still very exciting. Everything about Venus is exciting, and every couple of years we find something new about Venus, and I think it's about time that we send an astrobiology mission to Venus. It's super exciting. Phosphine has been discovered also, if I'm not mistaken, in our gaseous planets, in Saturn, and also in Jupiter. So, to sum it up, what we think about Venus and what we think about life. So, there is high pressure on Venus. There is a greenhouse effect going on. There's massive amount of carbon dioxide in the atmosphere, but none of these specific criteria has something to do against evolution of life. We know things that can withstand these conditions, fantastic. However, the problem is that there is really, really high temperature, okay? And there's no liquid water available for life forms. And every life forms on planet Earth needs to have available water to it. And on Venus, that's not happening. So again, Professor Charles Cockle had a very nice article called Life on Venus a few years ago. And he basically said that we're looking for some acidophilic, right, microorganisms that are really happy in acid conditions. They know how to enjoy the sulfate. There's the window open. They know how to enjoy the sulfate in the clouds, and they will live only in the clouds because that's the only place which is sort of like have normal temperature below 400 degrees perhaps. So it's a bit of an issue whether we will find life or not, but it's definitely a big puzzle. So thank you very much for Extremophiles, and now we can talk a little bit more and answer your questions. Thank you.
Okay, perfect. Can you stop sharing your screen, Reut? Yes. All right. Awesome. So we have quite a few questions today. So I'm starting with this one. Can you explain what a biosphere is?
Yes, the biosphere is all the things that living things live in. So everything, almost everywhere on planet Earth is part of our biosphere.
Okay, all right. That's a short and very specific answer. All right, because people here in the chat box were like explaining, some of them were explaining, and some of them were like, maybe there's different answer. Okay, cool. Then of course there was a question about Venus, but you explained it quite well. I also love the images that you show. That was like really, insider's view on Venus. So that was really cool. But with that, it's my personal question to you. Where else in our solar system you think these extremophiles might exist, or like has possibility that we will discover it on some other planets or satellites of some other planets in our solar system?
Yeah, so that's the 100 billion question, isn't it? I really think that follow the water was a wise decision by NASA from a research point of view. And I think the fact that we find water on Enceladus and beneath, and perhaps a salty ocean in Europa, Jupiter's moon, are very good target as well. I still think they could be life on Mars. I just think they will be underground in caves or lava tubes or underneath the caps. We know of life that are able to withstand hundreds and thousands and perhaps millions of years of evolution within caves. Caves are a wonderful, very natural habitat, very protects them from UV radiation and other things that are happening on the surface, and they are still able to work, to live there and have their energy and have food and nutrients. So caves are a big thing for me. Titan is an issue. Titan has lakes of methane, and while methane is a big biological signal, I can't imagine anything living underneath minus 230 degrees, which is basically, I mean, it's so far away from the sun.
Yeah.
It has very limited energy sources from the sun, and the surface temperature is cold. However, Titan does have an atmosphere and maybe does have an energetic core or some sort of an activity that would enable energy to any life form that's on the planet. So Titan needs more stuff to look at it. Yeah, definitely.
Yeah. Okay, awesome. So somebody has asked, this is an interesting question. Can humans be considered as extremophiles?
Can humans? No.
Well, in the beginning, even I thought maybe no, but just to discuss about it a little bit, maybe for some aliens, we might be extremophiles, like the aliens who would have methane as their natural habitat, which they're living in. Maybe we would be living in extreme conditions for them, so close to the star, or something like that. So I think it's more of a perspective. What exactly we call extremophiles, isn't it?
Absolutely. You can also claim that in the very future, someplace else where there are beings who are completely digital in their form, for us being in our physical form is considered extreme. So there you go.
Might be. Yeah, okay. Can you just quickly, like in just one or two sentences, can you fit in the definition of extremophiles? Maybe that will help understand the concept.
Extremophiles, there isn't, so extremophile depends on each condition. Like the temperature is extremophile, is for things that are able to withstand up to 70 degrees, and hyperthermal is whoever is able to grow and reproduce above 70 degrees. So extremophile, I would say, is very extremities of physical condition as we human define them. So we won't be able to live happily in 60 degrees, but a microorganism that will be termed extremophile. Same goes for high pressure, same goes for high levels of salinity, high concentrations of salinity, acidity, alkalinity. We know things. So for us to put our hand in something which is pH 1 basically means like, seeing your hand dissolving.
Yeah.
It's not good for us. But we do find microorganisms that has adapted, again, through the natural selection of Darwin and through so many centuries and millions of years to be able to withstand that. And that's pretty amazing. There's some fungi and lichen that can work and grow very slowly, but they do that in 3 pH. It's amazing.
Right. Okay. So connecting with this question, other person has asked that can we adopt ourselves to live or survive anywhere else in the space?
That's a really big question, isn't
Yeah,
it?
yeah.
It's a really big question. I mean, I think we probably can use technology much easier. We are much better at manipulating technology than we are manipulating our biology.
Right.
Our biology is a very complex system. It's like a chaotic system in a way. It doesn't work like an engineer would think because there's so many. It's an evolution of 4.5 billion years, right? So it doesn't work like engineering processes. So an engineer trying to make us work and live in space would sort of face it like an engineer when the process that brought us here was not really engineering anyhow. So I think we would be able to work and live in space. I think we'll first try to use the technology to our best of our knowledge, and maybe later on some interesting biological, but I wouldn't know which one.
Yeah, yeah, because somebody has mentioned it that a few humans have, broke the record by surviving in extreme conditions. So maybe they'd have better chance if they go to space or to extreme conditions. What do you think about it?
I think nothing on planet Earth is even close to what's happening in space. I think it's very, very hard in space. And I wouldn't recommend anyone trying just to go there. I mean, if you really want to go through the process of being an astronaut, you will be very well tested to
Yeah.
see how well you can survive that condition. It's very,
Yeah.
very hard. Yes.
Yeah, even like astronauts have to go through such hard training to survive in like completely tried and tested technologies or spaceships, and even that's so hard. So, just living in space for a long time. I think this is getting generated, these ideas are getting generated from movies like Interstellar, like a human being surviving through a black hole or extreme conditions like that, maybe.
Yeah.
But I think that's just better left for the movie right now.
Great movie. What can I say? Great
Yeah.
movie.
Okay, so Adarsh Kumar is asking, we know that long ago the average temperature on Earth was quite high. So do we have any thermophiles from that era surviving today, or are today's thermophiles completely new and different?
I can't say that off my hand. Basically, we do find very weird microorganisms. They're part of a big family, I would say not family, big class of organism, which we called Archaea. And Archaea seem to be very ancient in their design from some point of view. But so I wouldn't. So in some places, so for instance, one of the things you have to look at is, let's say we're talking about Yellowstone. Let's say we have isolated something from Yellowstone. I mean, how long has Yellowstone itself been on planet Earth? It hasn't been there for 4 billion years, Yellowstone. It might have been there for maybe 600 million years. So if I'm looking at a bacteria that I'd isolated from a place which is 600 million years old, then that bacteria or archaea in this instance is relatively new to that environment. That's what I think. And whether some of its characteristics are representing very old, then that could be. But still, if Yellowstone was there only 600 million years old, then the archaea its characteristic won't be very older than that, I would say. But it's a good question. It's a good question to ask.
Yeah,
Thank
really
you.
interesting. Okay. I think I'd ask the same question about the extremophiles, which are found in extreme ice cold condition on Earth. So, do you think that the extremophiles that we find on the poles of the Earth, we could find similar ones on the poles of Mars, maybe on the extreme icy conditions in the ice caps?
Yes, I do think that's a great question. Yes, I mean, some of the stuff that we find today in the poles of planet Earth, we know are bacteria which belong to very ancient bacteria that were here also in the beginning of planet Earth. And I'm talking mainly cyanobacteria and others. And if Mars has been like planet Earth, it had the same evolutionary trajectory, and at least was a very nice place, let's say around that 3.5 billion years ago, then yes, there is a chance that some of the stuff got preserved in its ice bowls and something is living there. Once again, you probably have to dig in, not on the surface.
Yeah, okay, got it. So Rupa Dhave is asking, do the extremophiles in Marina Trench have symbiotic relationship?
I think they do. I think some of the bacteria are living inside these lumps that we've seen. The worm, the hydrothermal vents worms. Sorry, and I think they have symbiotic relations. I think the worms give them a structure. I think they give them some nutrients, and also the bacteria are giving them also some sort of secondary metabolites in response. So yes, I think there is some symbiotic relationships going on. And the insects for sure have symbiotic relations, because I don't know if you're aware, but also cockroaches, for instance, their gut microbiota is completely symbiotic with the cockroach and provides. The reason cockroaches can eat almost anything is because they have this amazing gut microbiota, which do all the hard work of disintegrating all this stuff. So it's symbiosis is very common in the biosphere.
All right, okay. So again, Adarsh Kumar is asking us question, what makes tardigrades extremely resistant to high or low temperatures?
Okay, what's the question again?
What makes tardigrades extremely
Yes.
resistant to high or low temperatures? And in the continuation, he's asking, is it likely that tardigrades undergo a mutation that allows them to renew their cells frequently?
Is no, they don't go under frequent mutation, which allows their cells to grow or something like that. I at least have not encountered that one in research. Regarding how they are able to withstand high and low temperature, so they do have a lot of those heat shock proteins that I've mentioned before. That's one mechanism which allows for basically to roam around a cell, make sure that other proteins are still working very well. They have heat shock proteins working for them as well. I think they have some changes in how the proteins themselves are structured. They're much more compact in that manner. So if you have something which is fairly compact, it might still work under low temperature, but it also won't completely disintegrate when you have high temperature. And I think mostly the mechanism of being able to survive when you don't have water in your body, that's, I think, the main draw card here. Because if you have high temperature, the water evaporates and you're stuck without any water, just with vapor perhaps, but you're stuck without water. And if it's too cold, the water becomes ice crystal. And like we said, it causes mechanistic disruption to the tissues around it. So I think the main draw card here is that they're able to exert the water. The issue I would love to read about is like, how do they know they need to exert the water? Like, what is the environmental signal which tells them, okay, you're really in trouble now, just take out the water from your body? I don't know that. I would love to know that. I would love to know how quickly they can do it. I would love to know how it flows out of their body. Does it go through the mouth? Does it go through the other side? Does it go through their skin? It's amazing. So that's what I know about the mechanism of tardigrade. There are some great research articles in Google Scholar about tardigrades, and you're most welcome to look at them.
So it looks like there's a lot of opportunity in this particular regard, to understand lots about extremophiles or microorganisms per se.
Yeah.
So we have question from Atharva Tupe now. Could the signs of life be found on or near the sun's surface? Can the sun sustain life? Or I would say any star. Do you think any star could sustain life?
No, any star. So as you know, there's a lot of different stars out there, right?
Yeah.
So,
I mean, this
different
person
stars
has asked
have different
specifically
depth.
for the sun, but like I'd also like to extend the question for the other kind of stars as well, because knowing that the stars go through different temperatures throughout their life cycle. So sun is a middle-aged star, but what about the younger or older stars or white dwarfs and stuff like that?
Yeah, so I think that if you have a star which has lower temperature regime, then you're looking at something that might be able to support life. But at the moment, our life is not capable of supporting life on its surface.
So not any star.
I think some stars may be a white dwarf. Again, depending what in its life cycle where it is, there is a possibility that some part of its surface would be a good composite for life form, and things might actually work there. But we need to go and find out.
Yeah, it's even further than our solar system. We are still finding out life, or even like the possibility of life, not even life on Venus, which is
Right.
the neighboring planet. We call it sister planet to the Earth, and now we are talking about the stars, which are many light. Oh, that's Indian monsoon going on over here. So excuse
So
me.
that
Yeah, cannot
so
handle that noise.
yes, thank
All
you.
right. Yeah. So another question is, what is the oldest organism on the planet Earth now?
No idea. When you say oldest, do you think that there's something here that is like now alive, but it's like, I don't know, 100,000 years old? That's what you mean?
I don't know, but maybe something that we found, maybe like the first organism that we know so far, like that was the first thing. If I mean, anything would be okay for this answer. If something that is still alive or just the fossils, because I don't really know what exactly the
Yeah, I don't
question
know.
is.
I
Yeah,
don't know either. The question is not entirely not
yeah.
entirely clear to me. I mean, there is nothing now that lives that it's 100,000 years old. There's no such
Mm-hmm.
thing as far as I know. But we have evidence of life starting from 3.5 billion years old, but that's evidence. It's not the same thing.
Mm-hmm.
You can
Yeah,
watch my YouTube from last time. I think I talked a little bit about the
yeah,
origin of life, right?
yeah. You did.
So
Yeah.
that's so the answer is there. Thank
Okay,
you.
perfect. Also, one more question. It might be a little vague, but it's from my side. I'm just a little bit curious about the blue-green algae, which I heard is
Hmm.
extremely important in the evolution and beginning of the life. So,
Hmm.
can you just briefly tell me about it?
So blue green algae is the former name of cyanobacteria. It's the same thing. It's not an algae; it's a prokaryote. It doesn't have a nucleus. It usually has the DNA inside the cell. And cyanobacteria probably appeared between 3 to 3.5 billion years ago. And because they were the first life form to do photosynthesis, they changed the atmosphere of our planet. Before they arrived, we didn't have oxygen in the atmosphere. And after they arrived, we actually started to get a lot of oxygen in the atmosphere, up to the 21% that we happily enjoy today. And they changed the course of our planet forever. And cyanobacteria are a really big family of bacteria. They have many, many different traits, many photosynthesis capabilities, and they also live in many different environments. They're very tough bacteria, and they can be found in deserts, in polar regions, specifically Anabera, nostocs, and calothrix are very popular in the polar regions, both in the Arctic region and both in the South Pole. And you can find them living in symbiosis sometimes with lichens. So they do the photosynthesis, and lichens provide them with other nutrients and things.
Mm-hmm.
And it's a very successful life forms on planet Earth, and it's very diverse.
Okay.
So yeah.
All right, because I mean, I heard about it when I visited this lake, the Lonar Meteorite Impact Crater, which is located in Buldhana district in Maharashtra state.
Okay.
So I have also done a little bit video about it, but I didn't particularly talk about blue-green algae. But the lake has abundant blue-green algae in it, and it also changed its color to pink this summer. So I've made a small video about it. If you guys are interested, you can just go see it. And Reut, I'd love to talk about it separately with you and discuss more about it, learn more about it.
Sure. Sure.
Um,
Very interesting.
yeah. Okay. Are there any more questions? Can you see any more questions in the chat box that we haven't covered yet? Nirmal Gupta has written, as we found phosphine on Venus, is it possible that there may be other organic gas or life supporting minerals on that planet or exoplanets?
You mean on Venus or
Mm-hmm.
on exoplanets in general? Again, I'm not entirely sure. I
Uh,
mean, Yes, there is possible that there is other organic gas like what, methane. I don't think we found methane on Venus yet. I can check that out. There's many other gases. We haven't found anything substantial that we can say for sure that's something that comes from a life form on Venus. And when we're looking at exoplanets, it's pretty far away at the moment, but we're hoping that the next telescope, James Webb, would be able to help us understand the atmosphere of the exoplanets that were detected by Kepler and the TESS missions. And once we know, we will find exoplanets, but we will also be able to know what their atmospheric composition is, and we'll find out whether they have water in their atmosphere, whether they have oxygen. Again, oxygen is not a biomarker for life. I have to say that. We know there's a lot of life forms on planet Earth today who are happy without any oxygen whatsoever. So oxygen in the atmosphere is an interesting thing, no doubt about it. But it's not, in my opinion, it's not a strict biomarker. But yeah, in specifically on planet Earth, that oxygen was produced by cyanobacteria and by no other process. So for us, it is a very important biomarker in planet Earth. But again, doesn't mean that if you find a planet without oxygen, it doesn't mean that it doesn't have life.
Yeah, exactly. Yeah, I think that's like the massive takeout from this session or the overall astrobiology series, that it probably exists out there. My mentor who passed last week, Professor Govind Swarup, he always says this, and I'm remembering it again and again, that it is possible that life exists somewhere. The real question is there intelligent life out there? And that's what we are gonna think about a lot. Okay, then I guess those are all the questions for today. Thank you so much, Reut. That was an amazing session about extremophiles, which was really interesting.
Thank you.
Yeah, and
Thank you for staying with me.
yes,
Yeah.
yes, and everybody, this is a special video today because this is our 101 number video on our YouTube channel, and I'm so happy that with this series we have crossed 500 subscribers. We are up to 600 subscribers on YouTube. On our Instagram as well, we have been receiving an amazing response. We have 1,000 followers on Instagram. So I'm so glad that you guys are enjoying our content, and we'd be so happy to know what else you'd like to know more from us, what sessions you'd like us to do. The next session with Reut is going to be next Sunday evening, which is 27th of September, and we are going to talk about the technologies to detect the life. So I hope you guys are excited for that. If you have any questions for Reut, drop the comment in this video. If you want to have a certificate for this course, you are gonna find a link in the description of this video, which is a link for a Google form. So you can fill up that form, and we'll get back to you after the series has ended. And there'll be a little quiz before you get your certificate. So you can get certificate for this course, which is really exciting, and it's amazing how Dr. Reut is talking to us about astrology, astrobiology, and I'm just so excited with this session. So I hope to see you guys in the next session next week, same time. So clear skies until then. Reut, are you still there?
Yes, I am.
Okay, I'm just waiting for the YouTube thing to end so that I can end the stream. Whatever we are talking now won't go up there, hopefully. I'll end the stream before that.